Tobacco mosaic virus TMV. Introduction. Tobacco mosaic virus TMV is an RNA virus. It infects plants, especially tobacco and other members of the family Solanaceae. The disease causes great economic loss by reducing both the quality and quantity of tobacco crops. Its infection causes characteristics patterns. It causes mottling and discoloration on the leaves. Discovery. In 1886, Adolf Meyer point out the mosaic pattern symptoms on leaves of tobacco. The tobacco mosaic virus TMV was discovered by D. Ivanovsky in 1892. W. M. Stanley isolated it from infected plants and crystallized it. He found that the crystals were highly infectious. Symptoms. The symptoms are of different types showing various degree of chlorosis, mottling, curling, dwarfing and blistering of leaves, dwarfing of entire plant and discoloration of flowers. The variation takes place due to variation of virus strains and the variety of plants affected. Structure. It has rod-shaped structure with rough surface. Each rod is 300 nanometers long and 17 nanometers in diameter. Capsid. The capsid consists of 2,130 protein subunits called capsomers. The capsomers are closely packed and arranged helically forming a hollow cylinder. The central hole of the cylinder is 4 nanometers in diameter, which is throughout the rod. There are about 130 helical turns and each turn consists of about 16 1/3 capsomers, 49 subunits in 3 turns. Each capsomer is formed of 158 amino acids. Nucleic acid. Nucleic acid is one molecule of single-stranded RNA. It consists of 6,000 nucleotide pairs, 6,400 bases. RNA remains deeply embedded in protein subunits 5 nanometers from the outside. It does not occupy the whole. Transmission. TMV enters the host cells through mechanical wounds, by vector like Mysis persica, through infected pollen grain. Disease cycle. The virus initially infects wounded tissue of the seedling in seedbed or the transplanted in the field. After invading wounds, virus infects parenchymatous cells. The virus then multiplies and gradually moves from cell to cell through the plasmodesmata, and after reaching the phloem, it moves systematically and infects whole plant. Then, virus enters in cytoplasm where it stays in crystalline form. Disease management. There are following methods to control the disease. O. Physical method. Soil bed can be sterilized to reduce the attack and multiplication of viruses. O. Chemical method. By spraying the tannic acid O growing disease-resistant varieties. The best way to reduce the attack of virus on crops is the cultivation of disease-resistant varieties such as TMVRR2 and TMVRR3.